Hello everyone, my name is Ataberk Donmez, and today I will talk about my research titled stRainy, assembly-based metagenomic strain phasing using long reads. Starting off with background and motivation, we know that genome is the genetic material of a single individual, whereas metagenome is a collection of microbial genomes from an environment or a sample, and these samples are usually collected from places such as human gut, wastewater, soil, or marine. But why do people work on these samples that contain multiple species and individuals? Well, because metagenomics is a great way to study the composition and the diversity of these microbial communities. In fact, most bacteria cannot be isolated and cultured separately, thus metagenomics could be our only way to get their genome sequenced. Moreover, working on metagenomics can help us learn more about microbiome evolution dynamics, it can help us detect new pathogens, it can even help us create new bacteria-produced antibiotics. In fact, there is another biological challenge, and that is not only detecting different bacterial species, but also detecting different strains is equally important. And that is because different strains of the same bacterial species may show distinct phenotypes, such as different pathogenicity levels to humans, and different levels of antimicrobial resistance, and some strains of one species could even have some implications for cancer. So we have some very good reasons and motivations to study metagenomics data. So we get our sample sequence, we get a bunch of reads, long reads, short reads, doesn't matter. What is the next step? The next step is usually assembly, but there are some very specific challenges for metagenomic assembly, and these stem from the fact that we are trying to assemble many genomes simultaneously. And our samples not only contain different species, but they also contain different strains of the same species. And in our samples, we usually have uneven species composition, and this results in uneven read coverage. And last but not least, we usually see the same nearly identical pieces of DNAs in unrelated individuals, and this is called interspecies repeats. There are a number of long-read assembly tools, but only a few of them are specialized for metagenomes, namely ISMB and MetaFly. The first tool, ISMB, is designed to work only with PEG by hi-fi reads. The second tool, MetaFly, works with both PEG by hi-fi reads and Oxford Nanopore Technologies reads, but MetaFly cannot achieve strain level resolution in the resulting assembly graphs. So if we can't have strain level resolution in the assembly graph, what we do is we usually try to phase different strains, and there are different tools for phasing, but they usually work with diploid samples, and they will not work on metagenomic data. So to visualize the phasing problem, we try to look at the heterozygous regions with strain collapse unitics in our reads or references. And we try to assign these genes into their respective haplotypes. And this brings us to our tool, stRainy, because this is what we try to do as well. The whole pipeline of stRainy relies on something that we call informative SNPs. An informative SNP in this case is an SNP that separates a set of reads into two non-empty sets. So if you were to look at this figure, for example, let's say we have three different SNPs, SNP 1, 2, and 3, and let's say we know that we have 4 different strains. So looking at SNP 1 here, the two non-empty sets it creates is strain 1 versus strain 2, 3, and 4, because we observe this SNP in strain 1, but not the other 3 strains, and different subsets will be created for SNP 2 and SNP 3. And informative SNPs also can indicate phylogenetic relationships between different strains, and the corresponding phylogenetic relationship for these 4 strains based on these 3 SNPs are given in this phylogenetic tree. But how do we use these informative SNPs? Well, we use them to create a graph called strain graph. And the way it is constructed is that for every read in our unitic, we will create a node corresponding to that read. So in this toy example, we have 3 reads, so I created 3 corresponding nodes. And the connections between these nodes will be determined if certain conditions are met. The first condition is that the reads need to share a dovetail overlap. So in this example, we see that all 3 reads share a dovetail overlap, so the first condition is met. The second condition is that the overlapping region needs to span at least one informative SNP. Again, this is met for all three reads. The final condition is that this region should not have a non-matching informative SNP. So in this example, we see that the first and the second reads agree with this SNP, but the third read disagrees with the first two reads. So the connection is only between the corresponding nodes to the first and the second reads. So the third node will not be connected to any other nodes in this example. The next step after creating this strain graph is that we will use a community detection algorithm called label propagation to detect the different communities in our strain graph, and ideally different strains will correspond to different communities. Now let's see how the previous steps work on a larger data set. Remember that we repeat the previous steps for every strain collapse unitic in the original graph. And on the top figure here, we see the reads, all the reads in a strain collapse unitic. With the naked eye, we don't see any clustering tendencies, but if we create the strain graph, as we explained in the previous steps, and apply the label propagation algorithm, is that we will get such a graph where different colors correspond to different labels for each read. And then if we group the reads on IGV screen according to the labels coming from the label propagation algorithm, we will get such communities or groups on the IGV screen. Now, with the help of these labels or communities, we will create an overlap graph for each unitic. Initially, we have source and sink nodes corresponding to the beginning and end of the unitic. And we will have a node for each community. And these nodes will be connected if the communities overlap and if the consensus sequence of these communities generated using Fly are sufficiently smaller. And in the end, we will have a graph with multiple branches from source to sink. And each path in this graph will correspond to a different strain. So we will go back to the original assembly graph and replace the initial unitic with multiple new unitics. So here on the left-hand side, we see an assembly graph that is supposed to represent 3 different strains of E. coli. And in this figure, different colors represent different strains. So we see that a lot of strain collapse unitics in the left-hand side graph. And in fact, it only represents around 55% of the references of these 3 strains. However, if we apply stRainy to this graph, we see that we resolve most of the strain collapse unitics, and the represent length of the sequences increased from 55% to almost 93%. And moving on to the results, I will show you results with simulated and real data that contains both Oxford Nanopore reads and PacBio Hi-Fi reads, whereas they are evaluated using a tool called MetaQuast. Looking at simulated Oxford Nanopore results, we see that regardless of the number of strains in the dataset, stRainy produces the best results in terms of completeness, strain switch errors, and intra-strain misassemblies, whereas Strainberry has higher NGA50 scores. For simulated PacBio Hi-Fi reads, we can include Hi-Fi ASM Meta in this comparison as well. This time, we see that Hi-Fi ASM Meta and stRainy produce comparable results in completeness, strain switch errors, and intra-strain misassemblies. But Hi-Fi ASM Meta has higher NGA50 than stRainy. We see similar trends in real data, which comes from a mock community, and it includes 5 E. coli strains. For Oxford Nanopore reads, stRainy has simply the best results. For PacBio Hi-Fi reads, Hi-Fi ASM has slightly better results than stRainy, but stRainy is up there with it. In conclusion, we present stRainy, an algorithmic framework for long-read assembly-based phasing of bacterial strains. stRainy can operate on sequence graphs and therefore improve de novo assemblies or take advantage of pangenome reference graphs. stRainy is general enough to be applied to both PacBio Hi-Fi and Oxford Nanopore Technologies reads, which contain different error patterns. And finally, I would like to acknowledge the contributions of the people that worked on this project. Firstly, Ekaterina Kazantseva, who is the co-first author for this work, and my supervisors, Dr. Mihai Pop and Dr. Mihail Kolmogarov, and our group at National Cancer Institute as National Institutes of Health. And thank you very much for listening.